welcome to the meeting. It's like Susan's got 2 instances, Barshan has 1, I have 1. There you
I'm here.
go. All right, yeah. Hi, how are you?
Okay, except that I haven't done as much as I wanted to on projects this week.
Yeah.
I discovered a mess in my art drawer. Some certain rodents got in and got into the magenta.
Oh, no.
They must have got it all over themselves, but it got all over the artwork in the drawer. Yeah, mostly photographs, so they're replaceable. But
Yeah.
it's like, I have magenta toned artwork.
Oh, yeah, yeah.
Yeah. Anyways, I do have one paper. I think I told you about it, so I'll just put it in the chat, like
All
the
right.
reference.
All right.
So I'll do that. And otherwise, I haven't been doing anything.
Okay, yeah, well, that's okay. Yeah, why don't you go ahead and put that in the chat, and then maybe we'll go over it later or talk about it later. All right.
Okay.
It's like Hussein's here as well. Hello, Hussain. Are you, what's going on with you?
Hi, yeah, I'm fine.
Good.
Just been busy with some different things. Very, very busy with the CT project, with the RBY RCT. Okay, I've been working on a few other things with Dick, and as well as a personal manuscript I started writing myself on a theoretical biology framework. I briefly mentioned this in the Slack channel, and I was able to flesh it out a bit more. Essentially, I've come up with a new way certain complex systems can be under certain dynamics can be treated as stable given some conditions. And I know that sounds really, really generic and general and vague, but what I'm trying to do is come up with connections between forms of instability and some other theories in amplitude and entropy for cyclic systems that I've been observing across different areas.
Right.
I can talk more about it if you want, because this is something completely that I've more or less thought of myself and have now put into a paper. Yeah.
Yeah, that'd be great. You want to talk about it?
Sure. I mean, okay. I guess I can just start talking about it right now.
Okay.
No. Okay. Okay. So I used to play a lot of competitive video games, like these fighting video games, Super Smash Bros. Street Fighter, and you know, when you play these video games, there's a lot of times different players have their own style, the way the characters, fight each other. And one thing I noticed that was really interesting in some of the meta, some of the competitive strategies, was there was one point when at least there was some time when there were 3 people who had their own different strategy of playing that were all kind of together in this rock paper scissors like dynamic. One player would have a very, very control driven play style in which he would analyze the other player and predict your every move and be able to make the optimal or make the best decision. Yeah. There'd be another sort of player that was very, very, very aggressive. Okay, he'd always go for like the most powerful attacks at every single second, trying to just be as aggressive as possible. And there was another player who'd be very, very chaotic, very random, and very unpredictable. You know, he'd try to throw you off with different things. And for quite some time, these 3 players had themselves at each other's throats in this rock paper scissors type thing, in which the control player, so the one who relied on predictions and analyzing you, so we can call him control. He'd be very good at punishing and defeating the aggressive player, right? The aggressive player, we can call him pressure, yeah? The one who always goes for like the strongest one at every single second, yeah? He'd be very good at punishing the chaotic one, who's chaos. And the chaos, who was very, very unpredictable, was very good at punishing and defeating the control one, you know? Because you can tell if someone's behaving or acting in a way that's uncontrollable, then, you know, like you wouldn't be able to be predicted very easily either. Yeah. I thought there had to have been something that would explain this, okay, on some almost mathematical level. So I started doing research across different areas of work, like biology, chemistry, and among some other patterns I noticed, like in lizards of all examples, there's actually a species of lizards that has a very similar thing, where there are three colors of lizards, what do you call the botched lizard necks? Have you guys heard of these? The botched lizard necks, where there's orange, yellow, and blue. Orange lizards are aggressive. Blue lizards cooperate, like they work together. So, and they're all male, so they compete for female partners. The male orange lizards are aggressive. The blue-orange lizards are cooperative, or they're intelligent, they're strategic. And the yellow-orange lizards will mimic the actions of females and infiltrate, like, the harems and to confuse them, you know? So once again, you have these three poles, these three dynamics of pressure, chaos, and control. Okay? You can actually, so among the ways you can study these things, I was seeking to, first of all, figure out what is it that I'm talking about, right? Like, how would you describe this mathematically, yeah? For one thing, it's the system is non-transitive. So you know, like what the system is not non-transitive interactions that prevent a straight up hierarchy of, like, you know, there's no alpha male, there's no like the top monkey, the top gorilla gets more, you know, females than the others. With the lizards, they're all in a rock paper scissors, and they've been doing that for like hundreds of years or however long they've been doing it to secure partners and mates, right? This introduces what you call this non-transitive, anti-symmetric, cyclic dominance of behavioral patterns. And I wanted to mathematically describe or formulate what that simplex might be. How you might see if there's a pattern of control of tissue flow and gastro. Is this relevant too? Avian. Okay. Yes,
No, there's a paper I promised at the beginning.
Yeah. Okay. So yeah, I've now drafted this paper, and I'm going to submit it over to Dick pretty soon for his advice on things. But yeah, it's pretty damn thorough. I think it's at 33 pages. Yeah, I'm gonna have to trim this. I mean, you know, a lot of these pages are pretty short, but this is a 33-page Word document outlining how I've derived this. Essentially, it involves a Hough bifurcation. You can calculate a Jacobian. I performed the numerical simulations with calculating the instability, looking for how the entropy of the system. So once again, three poles: rock, paper, scissors, right? Pressure, chaos, and control. That's basically it. Yeah. And you know, I'm being careful. This is not like a new law of thermodynamics. It does not extend to all systems, you know. But it's particularly interesting that I think it can give you know, like not because I've already seen connections in it across different disciplines. In the lizards, you see this sort of behavior in economics between different economics models. You see this sort of behavior in certain types of embryogenesis. I think I need to look it up more specifically. So let me see if I can find the papers I was looking for. I don't know if it's cited in here. Oops. I'm looking this up on my computer. Yeah, it's you know, and also a bit of like a fiction type of metaphor for different stories and TV shows and things. Sometimes you see these archetypes in TV shows and movies where like you'll have characters that are very strategic. You call them like analytic characters, like masterminds. Maybe like, I don't know, like I don't know, like chess prodigies and stuff, right? And you see them in stories and movies and things where they think they can outpredict everybody, but they'll be thrown off by unpredictable environments. You know, you just have to be random in front of them, yeah. Or they get thrown off by chaos. And once again, a minimal entropy-based model, or a minimal cyclic replicator model with entropy-based fixation diagnostics. Yeah. So. And like I said before, I'm being very careful before just like claiming this is now like the new grand unified theory of everything, that this like explains all systems. But you know, what if the origin of life, for example, right, going from prebiotic to biotic species, from pure chemistry to cells and automata. What if it wasn't so much of hierarchy-based ranking, in which the best system beat out the less optimal systems, right? What if it was more so based off a rock paper scissors thing, where it just happened to be so that the balance was unstable, and one person that played in a different I keep playing rock and Bradley was playing paper, and that's how he beat me. He was able to figure out that I just kept it in one. And this is really peculiar and fascinating, not only for for the sake of the fact that it seems like it's much more balanced, much more stable than just straight up like Darwinian evolution, or just I don't know, biochemical expansion of more stable species over unstable species. If you the math isn't mathematical equations, I just need to go through this. But if you could showing that there is a hop bifurcation under which the systems change from one to another, showing that it follows the similar principles of rock, paper, scissors. These are all very, very, very strong directions to go in. I'm looking to probably submit this to mathematical biology, to the Journal of Theoretical Biology, or maybe chaos theory.
Oh yeah.
They're a good journal too, Yeah. So, we have the conservative system, the dissipative extension, and entropy expansion from entropy to instability, and a spatial extension. So the fixation scaling proposition. In certain microbial colonies, you see E. coli strains with these toxin sensitivities, yeah? And it's there's kind of these cycles between three things once again: toxin resistance and sensitivity, yeah? In E. coli. Yep. Sometimes with antibiotics, the three cycles, the three domains are antibiotic producing, antibiotic resistant, and antibiotic sensitive. So you see what I mean? Like one form may produce antibiotics, one form may be resistant, and the other may be very sensitive towards them, but also sort of like throw them off guard, like make things more unstable. So right, the dynamic was pressure, chaos, control. It's let me show the lizard. I'll get the lizards. I'll just share the screen with some of the lizards. It is so I know this is completely non sequitur, but these were based off discussions of various concepts I've been having with my meetings with Dick.
Yeah.
So that's where it came from. Yeah.
Oh yeah.
These are little lizard guys. Yep, they play rock, paper, scissors to win over mates. We got blue, orange, yellow. Yellow beats orange. Orange beats blue. Blue beats yellow. Yep. So once again, orange is aggressive. They're the bullies. They're the bulldogs. Yeah. Blue are the smart ones. They work together. They're cooperative. And yellow are the crazy ones. They disguise themselves as females and they mimic the females. Yeah. So they trick the other ones into infiltrating their harems. So that's kind of neat. Yeah. So cyclic dominance, we have an instability parameter epsilon, which corresponds to the environmental or the fitness type of asymmetry. And we show that entropy decline corresponds to the loss of behavioral diversity. That is very, very biologically meaningful. I don't know, maybe you see it in technology, like market dominance between companies, right?
Yeah.
You always have disruptive companies which are able to completely beat out the really manipulative controlling ones. But manipulative controlling ones are also able to stomp down on I don't know, entrepreneurs who are just way too aggressive, that's not gonna vibe in a lot of contexts, yeah.
Right.
So I think it's I can perform more simulations. I have my Python code, I have a repository. I can share the repository with you all. And once again, I first saw this in video games. You'd see in the video games these three different places: chaos and control. Yep. PCC dynamics. Pressure is massive force. Control is informational precision. Chaos is disruption. High energy, low entropy, reduction of local variance, or high Kolmogorov complexity, which is being unpredictable. Kolmogorov complexity, I can find that. Yeah. Interesting stuff. And I think the biggest thing I think that makes it stand out is that it isn't a single ranking. It's not a single hierarchy. It's non-transitive. So it's like one relationship doesn't define the other ones, you know. So like in a transitive relationship, like it's like A is bigger than B is bigger than C, so A is bigger than C. That's not true with rock, paper, scissors, and that's what makes it like really, really complicated, but also really fascinating, you know. I'm sure you can see this in mutation bias. Maybe like in video games too, like with balancing video games. If you ever play like Street Fighter or stuff like that, yeah. So the principle is that cyclic dominance occurs at the balance between this sort of entry based fixation. Okay, that's why we call it a entry based fixatedness. Yeah. Cyclic dominant. These are cyclic dominant systems which exist at the boundary between conservative entropy dynamics and dissipative entropy reducing collapse. Okay, yeah, those two things. Yeah. And of course, we can see where this can extend to other disciplines, maybe like fluid dynamics, maybe, you know, I guess control theory, maybe. You know, we want to see if it's robust, it can generalize, and see what other domains can pick up on it too. Yeah.
Well, that's great. That looks good, Hussain. Thanks for sharing that. There are a lot of applications. You know, you have, of course, the ones you mentioned. I'm sure there are others that where you have at least 3 agents. So you may have more agents, and really the key there is that it's non-transitive. That
Yep.
it's not that there's a hierarchy. It's sort of that you have this almost like feedback where, you know, it's self-regulating, and it's you know it's kind of like the predator-prey model, which is two-dimensional. This is 3-dimensional. You could be higher dimensional as well, where you have more agents in there.
Yeah, Ashby's good regulator theorem was one of the first ones I thought about.
Oh, okay, yeah.
Yeah. Ashby's good regulator theorem, which is the especially related to AI, to reinforcement learning. Every good regulator of a system must be there was something specific he said, like must be a model. It like must be a model of that system, yeah?
Yeah.
Ashby's good regulator, yeah. And this, you know, it is interesting because you like if I just show you this, there are 3 theorems, 3 propositions in my paper. You guys can read it. It's in the Slack channel, by the way. It's if you're in the URL,
Okay.
in breast cancer CT, in the breast cancer CT channel, just scroll up a bit, and it's like, I'll just post it again. Let's post it again. Do you want me to put it in like the DevoWorm or?
Yeah, if you want to.
Yeah, obviously DevoWorm and E. coli, like this is one of the biggest places you can see it. You know, and you see it in the markets in business. Okay, it's in general on OpenWorm. Okay, in the general channel on OpenWorm.
Yeah.
Let's give it a minute. Sometimes my computer gets slow. Yeah.
How would that relate to robotics, where if you have a robot that automatically explores its environment, it learns better and faster than one that stays within its environment and uses an epsilon greedy algorithm? Those ones don't learn as well as the ones that explore. That I don't know that much about robotics, but it sounds like it fits in with this, because that's the chaotic one that goes out and explores.
Yeah. Okay. So with robotics, typically with reinforcement learning, okay. Let's say I'm the instructor. I'm trying to teach an autonomous robot how to do something, whether it's like throwing a ball in a hoop, like playing basketball. Okay.
Yeah.
The 3 dynamics off the get-go would be control, what specific instructions and rules I give it with the boundaries. Pressure would be say how much energy, how much bandwidth, how much resources it have. Is it applying them in a way that's very, very forceful, or is it just like, it has half a tank of gas, right? And then chaos might be, what is it that I'm testing it on that could potentially deviate from the rules and norms? So I'm testing my robot to shoot a basketball in the hoop, but all the hoops are the same size. It's always from the same distance from the hoops. It's not going to be able to learn much because it's way too rigid with those rules and norms. So I could see this having relevance in robotics with that's just what sort of information you give it, how you map things too. Like to what extent, like, because you have to consider it in terms of like to what extent do these 3 cycles switch back and forth between each other? Yeah, does it make a legitimate rock paper scissors, or does it make something that's because so many models in scientific theory across areas like robotics, AI, evolution, they treat chaos as some external thing. Okay, you have to account for chaos, you have to account for entropy, you have to account for potential disruptions. The biggest thing about what I'm talking to you about that makes it completely different from all those things is that I treat chaos as its own principle, as its own pillar, just like the other two. That's what I'd like to see in reinforcement learning with robotics as well. So, like, chaos isn't something you minimize, chaos isn't something you need to avoid or not think about. Chaos is something that is integral to the system. So I would like to see maybe like multi-agent reinforcement learning. We can like simulate things using like whatever, like Python and stuff. Yeah. Like especially you would need many agents to observe these things. Okay, like not just me, not just you. You need many agents to see these sorts of things.
Yeah, I'm wondering if your chaos theory math might describe the tensegrities I'm working with too. But
Yeah.
people have tried, I think. Anyways.
You'll never guess because that competitive video game that where I first saw this, it was kind of interesting. I saw the commentators, they were talking about these boys who played competitive fighting video games. Okay, so people post them on YouTube and you make comments and stuff, right? And it's like the guy who would always be playing Control, who could just like basically predict your every move in a competitive video game. It was he's actually neurodivergent of all things. He has autism. And that was really interesting and unique to his playstyle, that he'd be very, very pattern recognition focused, and he would just write. He'd play these competitive fighting video games and study characters so deeply by writing all their like very precise, specific tech things in his physical notebooks. Like he'd have a pen and paper and just write all these things down about all the characters, and he could literally predict your every move no matter what you wanted to do. And he was so fine-tuned and on point that when people actually hacked the game, when they looked into the code, all of his physical notes completely matched how it was programmed. That is how. But you see his trade-off that you often noticed, and he was one of the best players they had in the scene, right? But he continuously lost against unpredictable players. He couldn't handle chaos. He couldn't handle chaotic play. Yeah, he couldn't handle chaos. And that's why, this is also why I always avoid chess. Chess is a terrible analogy. Like I've said many times before, don't look at chess. Chess doesn't tell you anything, Maybe you could look at competitive video games to teach you about these sorts of things. But like you don't see it in chess. You don't see it in chess. You see it in like real world things, Yeah, it's interesting stuff. You have to show it's cyclic, you have to show it's non-transitive, you have to show the transitions can occur. Like if there's some block preventing a robot from going from, say, a pressure state to a chaos state, then it's not going to hold true. Yeah.
Well, thank you, Hussain. That was great. Great discussion about that.
I don't know, like Batman, Joker is chaos, Batman is like pressure, maybe the police is control or something like that. Yeah, like maybe that's I don't know what like movies or TV shows I watch a lot of anime, so it's just like stupid stuff.
Yeah.
Because like it is interesting. Like if you think about Batman, right? Like, but obviously Joker is like he's violent and crazy and stuff. But Joker thinks of himself as society, and he sees Batman as the antagonist. Like what would things look like if you saw chaos as part of the system in and of itself? that's something you don't see it very often in these sorts of things. So
Yeah. All right. Well, why don't we move on? I did actually want to talk about the laws of the game, which actually I brought up in a meeting last year. And there's a paper, I think it may have been Hussain who posted it. It's the laws of the game at 50. And so it's
yeah, I think I remember that one. It was
Yeah.
by that paper. Yeah, Laws of the Game. So once again, Laws of the Game, is the it's by Winkler, right?
Winkler and Eigen. Yeah.
I think I have it somewhere. It's in my room here. I don't know.
Yeah.
A while ago. But yeah, so 50 years later, because I guess it was last year because it was published in 1975. Yeah, so it must have been 50 years after Eigen and I can show the paper right now. You want me to show my screen?
Yeah, go ahead.
Because yep. I think I posted this. Oops. Sorry, my computer just got slow. Okay, thank you. Symmetry breaking, this one, yeah?
Yeah, that's it.
And this is with Savino from Italy.
Yeah.
Cool stuff.
Yeah. So so this just kind of goes over this book at 50 years, and they introduced these bead games in the book. So the bead games are here on the right. Could you click on one of the bead games?
Yeah. Oops.
Yeah, so that's a board game.
Yeah. So what rules, what laws, what norms go on with symmetry? Like this, it looks a bit like Othello. You guys have played Othello, maybe, or like backgammon, Othello, a bit like that. Yeah.
Yeah.
A random walk. So this game is called equilibrium. That's what they call it scientifically. So how do the principles of nature govern the rules of this game? That's basically what people are asking. Yeah.
Right.
Can you see board games as like a diffusion process, the same way you see cells dividing or cells diffusing? I stakes rock paper scissors. Yeah. We always think of rock paper scissors as completely random, you know.
Right.
But if there's a logic behind it, you know, I mean, it is random. If you and I played, it is, you know. But in the real world, if there's like a legit logic behind it, then there is that, you know. Yeah. So.
And I think the important thing is that they were using games to model systems, and that comes, of course, from game theory, which, of course, von Neumann is known sort of as being the first person to propose. Although I think people would have kind of hit on it independently. But this is like, where you say, okay, this game represents a system, and so you have to map all the behaviors to the game. And and sometimes it works out well, and sometimes maybe not so well. It's kind of one of these problems where, like, these bead games are very good at modeling molecular phenomena. So you had different types of protein problems, or problems with a random walk through like a genetic space, and you can solve those, chemical kinetics, things like that. And then, of course, you brought up animal behavior, like lizard behaviors, and those models that as well. Although you have to be more selective in those, into what it's modeling. So, but I think this is pretty interesting, kind of reflecting on what people are doing. Last year, I think it was the October 20th meeting, I went over some of the things that people have done with this, and I pointed out this website where this guy had written some programs that were based on laws of the game. So he like programmed out some of the bead games, and I think it's available on GitHub. So that's that's available. And so yeah, people have worked out some of this stuff beyond like what Eigen and Winkler put in the book. And I mean, game theory is used pretty commonly. And there are interesting connections between game theory and like reinforcement learning too. So you have like reinforcement learning, you're looking for policies, optimal policies, and that kind of maps from like strategy suites and optimal strategies and things like that. So it's very,
Yeah,
very,
yeah. So once again, what if the optimal strategy isn't always about being the best? What if the optimal strategy isn't always about having the most resources, having the best fitness state? What if the optimal strategy, is much more complicated? Yeah.
yeah,
That it depends on what the people around you are doing. That it depends on, it depends less and less on whether or not you're the best or you're the worst, and more on just something more complicated.
yeah. There are also some interesting connections with information theory because, a lot of information, a lot of games require information, or sometimes they require perfect information. Sometimes players have imperfect information, and
The fact
sometimes,
that I can tell you about the competitive video games where somebody who memorizes every detail of every fighting character in the video game still isn't the best.
yeah,
You can still beat them by being unpredictable,
yeah,
So
variation versus like regularity,
yeah.
which is and then so this is an interesting paper. If you go down more, so they're actually simulating things here, and they're showing the spontaneous symmetry breaking, which is actually something, of course, in embryos that we're interested in with symmetry breaking. So, one of the things about embryogenesis is you get these symmetry breaking events where you get, you have symmetry and then you have maybe like an asymmetric cell division, or you get differentiation into a tissue. And so you have these events that like create structure. And you think about it like an embryo is this thing that is in the one cell state totally symmetrical. That means there's more or less invariance at the cellular level. At the molecular level, at the cytoplasm level, maybe not. But we've talked about this with some of the things that we've been talking over the past few weeks. But that you get a series of symmetry breaking events where you get like an anterior end and a posterior end, or you get some like structure with respect to organs or tissues, or asymmetries between left and right. And so that's how a lot of people have tried to think about embryogenesis, but they've never kind of put together. I mean, I guess people have tried to put together formal symmetry breaking.
Yeah, because it isn't just rock paper scissors. It's not for any rock paper scissors dynamic too.
Yeah.
Like you actually have to formalize and show how does, say, a more aggressive dynamic beats a more unpredictable one, which should in turn also beat a controlling one, which should also beat the aggressive one.
Yeah.
To what extent can they be balanced? Is it always in that direction? Like, does it ever flip? Because if it's cyclic, it can be non-transitive. Then there could be situations in which, it doesn't always go through as easily. Yeah.
Yeah.
So it's just like that. Yeah. So if this gets published, it could lead to maybe many other papers off of this. We could see how it branches into other disciplines and maybe become like a staple of game theory and information theory too. So maybe.
Yeah, that'd be great.
The more like casual term for this would be PCC, pressure chaos control. So just call it, yeah.
Great.
Yeah.
So thank you, Hussain. So I know Barshan last time. I don't think he was able to present on his stuff too much, but he wanted to present something, I think. Am I correct?
No, I presented my last PPT and I also submitted gave a paper idea.
Okay.
And I'm working on that. I'm searching for the data sets available, and I went through the past projects and I looked through the data sets and I analyze them, how I can integrate those into the concept of the edge set attention, and I'm still working on that.
Okay.
Yeah.
That's good. Yeah, I guess you did present. It's just that I thought you maybe wanted to present more because you did put your slides in the Slack.
Yeah. That was on the Eigen vector, Eigenworm and stuff.
Okay, yeah. All right, thank you. I don't know if Shreyas wanted to present today too, or
Yeah, I
have a short presentation. Like
Okay.
I have some ideas I wanted to present. So yeah, I'll just start now.
Yeah, go ahead.
Yeah, okay. So I'll start off. So as just a short recap. So last time up until now what I've done is for so the topic is like of course interpolation of connectome. So this time I have done some literature review and experiments to biologically ground my methods as to if they actually make sense or not. So a short recap. First time I implemented a GCN to get the latent representation of neurons, and then over that I used a plain neural ODE solver. And then later, last meeting, I put like transformers into it, GATs, and with a neural ODE solver plane, and then a biological bias regularization terms into it in the loss function. So I got results which did correspond with some paper findings, and yeah. So this time what I aim to do is finding some biological constraints from literature. Like these are some of the findings which I'll come to, and like one by one I'll come to, like contact scaffolding, Hough dynamics, motive evolution, and all. Then some validation strategy. So one problem which I encountered is that okay, how much ever models I make, like how much ever methods I try, it's difficult to understand if the interpolation which I'm trying to do actually does make sense or not, or if the structure is like, does it actually correspond to some brain structure, or is it just minimizing the loss function, or is it actually getting or are we actually getting some functional structure? So what we can do is so we can do functional check using a paper which I've cited. It's basically it maps the entire signal propagation atlas to predict the evoked kernels of C. elegans. So I'll take through that, and then at end we can do ablation to show why biological priors matters. So yeah, I'll come to all of these 3 points: structural checks, functional checks, and ablation. And now the methods which I'm thinking now is like some sort of ODE plus GNN. Now why do I write here some sort of ODE? Because I'm thinking about hyperbolic spaces where we can naturally model trees. Since the network is not a linear network, it's a tree sort of network which grows exponentially. So that's why I'm thinking on these lines. That's why I've written some sort of ODE solver plus a GNN with some attention into it to create a latent space. And the second thing is a differentiable axon growth hybrid, which I'll explain. So this is the executive summary, which I intend to present today. So this is just a short recap of this paper from which I implemented biological biases last meeting. So these are just like the like physical contact increases, like it's a strong constraint where the neurons tend to get connected to the ones which they are physically close to and contact with. So another thing is like as development happens, structures become symmetrical, more and more symmetrical. Symmetry, I mean right-left symmetry. So if I cut it transversely, I get symmetrical structures. Formation happens, modular structure like feed-forward fraction, as we discussed last meeting, like what is feed-forward signal flow. And then there are 3 types of edges. So this is just a short recap. Yeah, so like starting with the presentation. So this is a paper called like Dynamic Axon Growth Generative Model, where a simple geometric driven axon growth can I just show the paper, or like I have prepared slides, but I would be more comfortable like showing through the paper. Is it okay if I show the paper?
Yes.
Yeah, just a sec. I'll just show the paper once, entire screen. Share. Yeah. So yeah, it's this paper. So they are it's a generative model to model connectome dynamically. So what's different here is that this is something different from the method which I have been trying up until now. So up until now, I've been trying like deep learning methods, applying graph neural networks and all. But what they're doing basically is, so what they're saying is that connectome-like networks have been generalized into silico models. Just a second. Yeah, for like connectome-like networks have been used for certain organisms, which includes C. elegans. So what they say is like axonal growth and guidance are important mechanisms that shape the brain. And yeah, so this is the important part where this says each brain region exerts a distance-dependent attractive force on an extending axon step. So like assume I have an axon. Axon is basically for neuron. What is the end part which connects to the next neuron is the axon. So the extending axon step forces are distant dependent forces are applied through the neighboring regions of brain, steering the direction of axon growth. So this emulates the process through which axon growth cones react to molecular guiding cues through concentration decays with distance to chemical release sites. So what this basically means is that the ones which are closer to the axon applies more force, and ones which are farther applies less force. Like it's just plainly that. Then if we come to this is how they model the force, where this is like I equal to 1 to n, n is the like number of total neurons which I have, and Ri is the coordinate of ith node center. Then so here's the important part. So the model is governed by two basic key parameters. One, beta is the distance dependent decay of attractive force, and ls is the length of each extending step. So basically length of axon, like how much it is extending and connecting. So beta emphasizes the guidance from local adjacent nodes. Like basically beta just shows how much is the attractive force. And coming to like the important findings, I'll just keep it short, not too in the depth. So the results which they got suggested the combination of beta and ls. So the combination of beta and L generates connectome with realistic properties. So what properties do they quote? Is that so scale-free degree distribution. So the distributions of the connectomes are in a scale-free manner, like scale-free degree distributions, brain-like axonal bundles, negatively correlated connection weights and distances, and log-normal distributed weights. So weights, they mean, let's say there is a connection between neuron A and B. So how much is the weight? How many times are they connected? So the weights were normally like log normally distributed, and the strong range connections that deviate from EDR. So EDR basically is a method which just models how connection will be, which fails. EDR is a method which they have quoted in previous papers. So which fails to model long range connection. It says that okay, if there are connections, they'll be short ranged, and some other neuron on the other side won't attract neuron on the other side. Whereas this method, if we tune parameters, like if we get optimal beta and L parameters, long range connections can be modeled. So like variations in B had a stronger effect on degree distribution, and changes in L were more closely related to weight distributions. So this is a general method. So I'll just show what does the circle diagram mean. So here they have a circle which is the boundary of the neuron. Like here my like basically this is my simulation world, and here there are neurons situated on the edges and they connect to each other. Like right now they haven't put a like entire 3D space, like it's a 3D space, but they haven't put neurons in the middle. They have put it on the edges and they are simulating on a smaller scale, and it works, and it is coinciding with the results which are expected. So so going back to this, so this was the first. So this is just a slide of it. So each step I'll just go ahead. So this is the variation which they saw as beta and L decrease. Like I also just replicated their findings as to see if it works. So as beta and L were varied, these are the findings. So optimally, so this is the like optimal beta was 0.992 and LS was 1.992. So around these values, the structures were coinciding with the results which were obtained. So this is an interesting idea which I felt, which I aim to use more in C. elegans, is because this is something which is more biologically driven, and like it has some basis to which we can rely to. Okay, any questions for this?
Yeah, I think that's interesting. If you go back to that last slide you showed.
This one?
Yeah. So what is beta and what is L sub S? What do those represent?
Yeah. So beta is the parameter which, as you can see here in the formula here, beta is the parameter which models the attractive force between neurons, between the wait, I'll just highlight beta is a distance dependent decay of attractive force. So as it's exponential decay, you can think of it sort of exponential decay, where as my neurons are distant farther, say I have a neuron A and a neuron B. If distance between A and B is quite large, so the beta parameter would be low, and then if my ls is the length of each extending step. So if now I have a neuron A, how much can it extend, right? So those are my parameters which I'm setting, which depends on the setting. So if I keep setting such that I have beta as 0.992 and ls as 1.992, then this results are replicated.
Okay.
So basically, beta is a parameter which represents attractiveness, which represents the force upon a particular axon which is exerted by neighboring axons, and ls is the length by which the current axon will extend and connect to its neighboring axon.
Right.
Yeah.
And so this is then this next slide, go forward one.
This one?
Yeah, that one. So this is the dynamic axon growth generative model. So this is like sort of figuring out how cells connect together and kind of working backwards. Yeah.
Right.
Okay.
So this model, they explicitly state. Yeah. Sorry. Continue.
Go ahead. No, go ahead.
Yeah. So this model explicitly stated that since they didn't want to go towards a more AI model where things are uninterpretable and some things results do come out but we cannot interpret it. So they tried to put a simple geometric driven interpretable model like mechanistic interpretability where they can understand like how things are rolling out. So what I aim to do with this is like I aim to incorporate this into my graph neural network model where I can have some sort of groundedness for whatever results I get and I can have some interpretability of my model.
Okay. Yeah, that's good. Yeah, I like the network topologies here. So this is just a range of like if you vary the values, you get different types of connectivity. Have you looked at the network statistics on these, or?
Sorry, I didn't.
Have you looked at the network statistics on these, or what?
Statistics as in I didn't get you.
Well, like centrality and other types of things. Do you know if like these networks exhibit small world properties or other types of things? I know it's scale free.
I do remember them mentioning small world properties in the paper. I do remember reading it. I myself haven't done this.
Yeah.
World. Just a second.
Yeah.
Okay, maybe not. I'll just look it up if it is there.
Yeah.
I'll just see it once. As to the statistics of the model, I'll look and update.
Well, that's fine. Yeah, I'm just curious. But yeah, it looks like it's so these are generated from the data or from. Yeah.
So okay, this data which they used in paper is the human brain data, but as they specified in the start, this can be applied for any organism like C. elegans, humans, any like say dragon, like I guess some butterfly they experimented something, but yeah, so this can be experimented for any sort of neuron, not specific to any organism. This is just a property of neuron, like of axon that it grows in a certain way. So they tried to model the axon rather than the connectome of a particular organism through which they can generalize it across organisms.
Okay, it sounds good. Yeah.
Yeah. Also, I'll move to the next thing. So yeah, this was something which is the post-embryonic remodeling of C. elegans motor circuit. Also, one thing I'm presenting a bunch of different ideas. Then in the end, I'll just link everything and I'll show what I aim to do with all these ideas.
Yeah.
These are like bunch of discrete ideas which I'm presenting at first. So yeah, this is a paper called post-embryonic remodeling of C. elegans motor circuit. So particularly for motor circuit. So what it focuses on is specifically on the motor circuit during the post-embryonic development. So as you know, there are like 3 stages of development. L0, L1, L2, L3. So approximate L2 is, as you can see in the figure here, L1, L2, L3 are sort of the post-embryonic phase. The juvenile wiring happens in the L0 phase. So after that, the new motor, what they say is like the new motor neurons form synapses before old ones fully lose theirs. So if I have a motor circuit, in that I have some old synapses and they're evolving into new synapses. So the new synapses and old synapses would exist together for some period, for like some instances, they would exist together, and then the old ones would be released. So basically, the rewiring progresses in a spatially ordered manner, like in the anterior to posterior, and the functional. Yeah. So basically, the point is that the transition. So just a second, I was getting a call. Yeah. So there are transition phases where two partially redundant circuits coexist, right? So why this is important? Because here I'm trying to model. Here I'm trying to interpolate the connectome which we have. So again, the basic goal is to interpolate connections and interpolate the connectome. So now, if I want to interpolate connectome, there will be some transition phases where the two particularly redundant circuits would coexist. So this is what I got to know from this paper, that okay, I need to take into account like the connections shouldn't be abruptly cut. They should be evolved in a manner where I'll have the new connections and old connections in there like at the same time, and then the old connections would be set loose. And one more thing which they identified was that the functional motor output always remains stable during the structural change. Even though I'm going through a structural change, the output of the motor circuit wouldn't change. So why was this? Because there was a functional stability constraint is there. The hub neurons which are there, the hub neurons of the motor circuit, they do not change. The wirings through them do not change. That's why the functional motor circuit remains stable, and when it evolves, it doesn't affect the function of it. So as you can see here in the figure, synapse formations are happening along with outgrowth, along with synapse dissembling. So synapse dissemblings, if you see the small triangle here at the bottom, so that starts after a while synapse formation starts. So yeah, this is the graph, like the pictorial representation of what I'm trying to convey here. So yeah, this is the broader point which is useful for my goal. Of course, there are a bunch of other things in the paper as well, but these are the things which I think would be relevant for me. So yeah, any questions on this thing?
No, that looks good. So we have neurogenesis and then outgrowth, synapse formation, synapse disassembly. Basically, as the synapses are recycling, they don't just, there's no switch. It just kind of, you get one set of synapses and then another set of synapses, and then the first set dies off and then, or disconnects, and then the second set takes over, and it allows for smooth output. So you can have that kind of plasticity without disruption in behavior. Yeah, that's
Exactly.
That's cool.
Exactly.
And so that would be in your network. That would have to be accounted for. You plan to account for it.
Yeah. So I do plan to account it through like just whenever I remove a connection, as in if to put in mathematical terms, there are vector embeddings for each connection. So whenever I aim to remove a vector embedding, I will delay the removal. And when I add, and then after, well, I need to figure out when do I, like, how much delay should be there. I need to figure out that aspect. So I'm thinking on that thing. But what I aim to do is I'll delay the removal of a vector from my network. So yeah, I just went back to my network which I had implemented previously, the GATs along with neural ODE, which I showed in last meetings. So I did see that there were like abrupt changes in network which shouldn't be there according to this paper. So yeah, this paper helped me to understand that okay, it shouldn't be abrupt changes. I'll like model it accordingly. So yeah, coming to the next paper, which is a signal propagation atlas. So this is a way to validate basically. So what I aim to do is through this, basically I just tell what this paper is doing. So this gives a measured functional, this gives a measured functional atlas. It gives sign, amplitude, time course of signal propagation. So this paper has nothing to do with the anatomy. So why am I looking at this paper first of all? So the aim of looking at this paper is to validate my anatomy. So if my anatomy actually makes sense, I don't want to make a model which just minimizes loss on my time steps. So currently what data I'm using is through the Vivlet paper, the Nature Vivlet paper, which has like 8 time steps data, and I'm fitting my model through it. But it shouldn't be something which I'm just fitting on 8 time steps, and the rest, like interpolated points, are not making sense. I want to build something which does make sense and which actually works. So how will I know if something actually works is through this paper. As to this paper has mapped the signal propagation through the C. elegans connectome. If I fire a particular neuron, how does it affect on other neurons is what they have done. I'll just show it here. Where have they done it? Just a second. Yeah, so this is basically what they have done. So yeah, this is the paper. So they have systematically measured signal propagation in around 23,000 pairs of neuron across the head of C. elegans, and they measure the sign, if it's inhibitor, excitatory, strength, temporal properties, and so on. So of effect of, like, if I fire one neuron, how does it have the effect on other neurons? So this is what they are modeling, and this is their the whole picture of, like, how each, if I have to interpret it, I'll see, like, if I fire ADER, how effect does it have on the rest of them. So, like, rest of them are listed here bottom. So how each of the firing has effect on each other. So this is something on Q value, which they developed a statistic framework to describe to identify neuron pairs that can be deemed functionally connected if Q value is for a certain value. So if firing of one neuron has effect on another, is basically what it means. It's functionally connected or not. So they're modeling that. And yeah, it's like quite elaborate paper, and they have many things. But this is I'm not using this paper for the functional part of it. I'm using it to verify if my connectome actually makes sense, and if it's functional. So there's no use of my interpolated connectome if it doesn't work like a brain, or if it doesn't function right. So that's the reason I'm using this. So we can use it as a primary functional validation corpus, as currently it's difficult to verify the correctness of interpolated graphs based on just visualization. So last meeting I showed visualization, but it wasn't quite interpretable. Like, okay, one node is appearing, something is disappearing, but I can't make anything out of it. So I'll compare predictive effective propagation from my interpolated graph to the atlas. So one thing which we can do is we can, like, optionally train a small mapping from anatomy to functional kernel and show that the interpolated graphs produce the kernels. Like, I'll train this a small mapping on my interpolated graphs, and I show, and hopefully it will come out to be that the kernels which I find are closer to Randy's measurements. Yeah, so just that. And so the validation metrics will be based on Randy's metrics which I've done. So these are a bunch of mathematical terms like kernel explained variance, kernel time to peak errors, and so on. So not going into the exact technicals of it. So yeah, this is what I aim to do through this. Yeah, I guess, yeah, this is all about it. Any questions on this part?
No, I don't think so. You're just validating the topology, you're saying, is it?
Yeah, right. So this is a way, like, I think, like, I'm not pretty sure because I haven't seen this way to validate the topology. Like, one question actually I had, like, does this make sense? Like, can this be a way to validate? Because I didn't find it somewhere. I just thought, okay, this can be when I was just looking up for papers related to the work which I'm doing. So this paper came up and I read. So, like, does this make sense? Is this a good way to validate my topology?
Well, I guess so. Wouldn't you need some data, functional data, to do it? Of course. Do you have a data set in mind, or okay?
Yeah, so they have this data set, functional data set, entire data set as to how action-reaction pairs occur.
Okay, that's good. Yeah.
Okay, so yeah, I showed the figures from the previous paper. Again, so another way to validate, which I found in a paper, was that it's basically the motif fingerprint. So what is it? So biology is built on repeated subcircuits called network motifs, as shown here. Like, one of the motifs is left-right symmetry. Motif is basically something which occurs many times in a particular setting. So setting R is a connectome. So if I'm getting this type of left-right symmetry, so this is a motif. If I'm getting this type of, like, connectivity motif, like this 3 neurons are connected in a particular way and it's occurring multiple times in my network, that is a motif. If I'm getting this type of hub neurons where I have a bunch of hub neurons and there are branches from each hub neuron, and the branches in themselves do not interact with each other, and this pattern occurs many times in my network, this is a motif. So this is what a motif is. So the test, I'll count the distribution of these motives. Like say 3-node motives is what is this, like 3-node motive in the ground truth versus my prediction. So why a random graph can match the degree of distribution, like number of edges, what connects to what, but it will fail to match the motive distribution. So if my model captures the feed-forward loop motive or like these motives, then actually it has then maybe it has actually learned the brain logic, is what we can hope for. So yeah, this is one of the way which I think can be a good way to validate. So yeah, any thoughts on this?
Okay. I mean, yeah. I mean, it does reproduce the behaviors. I guess, yeah. I mean, it could learn the logic of a complex system too. But I guess the idea is that it's not at chance, which is usually the way they validate networks. They say it's a random network, or is it like structured in some way, and that's really what you want. And so yeah, that's good.
Okay. So yeah, this was all the papers which I reviewed. Now one last thing which I'll come to is one of the bottlenecks which I felt again, like this week I worked more upon just analyzing what I did in my past 3 weeks and how does it make sense. One of the issues which I found was like there's a bottleneck in the Euclidean space, as to it cannot represent trees and like connectedness and network in a very efficient way. So what's the problem is like biological networks are trees, not grids. So my neural development creates branches, hierarchies, and centralized command structures like hubs, which we saw here in the hub neurons and all. So why does Euclidean fail? Is that it's a standard flat space and the volume grows polynomially, like exponentially polynomially. So to embed an exponentially growing tree without crushing nodes together, you need a massive dimension. Like in a Euclidean space, I'll need like a large dimensional space, which introduces noise, overfitting, and a lot of issues. So how I just reviewed on Google, like how can I solve this issue, like and all. So I encountered like there's a hyperbolic space which is a solution for it. So it grows, the space itself grows exponentially. It mirrors the natural expansion of biological trees. So there's something called Poincare ball model, where we utilize Poincare ball model to embed the connectome with minimal distortion in lower dimensions. So like so dimension maximum, it uses like 5 dimension can represent. So we can't visualize like 5 dimension. I have visualized a ball here. I'll just come to it, but we can't visualize 5 dimensions, of course. So what is biological representation of coordinates is like basically there are 2 things called radius. So it encodes the hierarchy, the hub neurons. Hub neurons again coming to red ones are the hub neurons. The black ones are its subsidiaries. So hub neurons are pulled to the center, and the sensory muscles, which are the other neurons, are pushed to the boundaries. And one thing I guess I missed to show here is like the hierarchy, which I mean there are hierarchies in the network. Say sensory is connected to inter is connected to my motor neuron, and so on and so on. So those are the hierarchies which I'm trying to model. So the sensory and muscles will be pushed to the edges, like a larger radius, and hub will be center. So the angle theta encodes the function. So neurons with similar roles, like say motor circuits, cluster in a similar angular sector, even if far apart in physical spaces. So this is how it efficiently models the hierarchies, right? So say this is, I just like this is not an actual Poincare ball or something. So like Matplotlib, and I just modeled it. So just I can show something. I'll say this is my ball. Like you can imagine in 3D space. So like red ones are hubs. These are the inters, and green ones are the sensory / motors. So my connection, geodesic connection, can work in this way, right? So I can represent it efficiently in a Poincare ball or disk. So one thing, so like this was the setting, basically what is the setting. So now how will be the generative interpolation via geodesic flows. Now again, what is geodesic? One thing to point here. So geodesic is the distance on a globe, you can say, like how distances measured on Earth are like geodesic distance over a curved space, basically. So one thing, like standard thing which we can do, which I'll try at first, is the hyperneuron ODE, which is I'll have canonical positions as I showed in last meeting, like through OpenWorms paper. I had all the positioning like in 3D. I had the positions of each neuron, which I borrowed from OpenWorm paper. Canonical positions are projected into hyperbolic tangent spaces, and why tangent? I'll come to why I'm saying tangent space. So the ODE, the dynamic ODE, so instead of the unstable operations on a curve manifold, so operations on a curve manifold is quite unstable. So we evolve a system into the local tangent space using a neural ODE. So we'll project it into a tangent space. We'll work neural ODE in that space. So neural ODE is just this like z dz by d theta z is the latent representation which I'll get from my graph neural network. F of theta, which is z and t with the parameter. So this learns the vector field development. In the exponential map, the evolved state is projected back onto the Poincare fold manifold from the tangent space at each time step. So one new thing which we can see, I just did some literature review again, and I found like one linear versus geodesic. So the standard interpolation draws a straight line between developmental stages, and which is a biological false, which is not a straight line. So our model computes the geodesic, the optimal curve on the manifold, like the optimal curve through the network. Like there's not a straight line. So actual network is also sort of a 3D geodesic, not a like just a flat straight line or something. So the optimal curve on the manifold representing the most efficient developmental path. This is a mathematical function representing it, not to go in the exact nomenclature of it. So like how will the decoding done? So the connectivity probabilities are reconstructed using something called Fermi-Dirac function based on the hyperbolic distances to ensure predicted graph retains the Ridge Club as in the connections, the hub basically, the Ridge Club hub structure observed in so and so paper. So yeah, this is a new thing which I'm trying. Currently, it's like I haven't attached results because I'm reading the theory of it and I'm trying to get the math sorted out and I'm running it. So this is something new which I'm trying out on a mathematical side. And the previously one were the biological basis which I need, because currently my models, okay, maybe the loss can be low, but it doesn't actually represent the biology which I wanted to represent. So these are helping me to ground my models on the biology, and this is something which I'm trying on the mathematical side and trying to get a better, like efficient way to build my model. So yeah, this is it from my side for today.
Yeah, I wanted to mention that the Poincare disk is a hyperbolic space, and like if you think about the embryo, because it's curved, using a hyperbolic space is a little bit more, I guess, gives you a better handle on that space. Using Euclidean distances is fine, but it doesn't capture sort of the way. It's like using Euclidean distance on the surface of the Earth, measuring a distance between 2 points. You can get that distance pretty easily, but that's not like if you want to fly a plane from point A to point B, you would take like a curved path because it's actually shorter. And so that's kind of the better account of the embryo, better account of the connectome, because the connectome is in this curved space as well. So that's good that you have that. And a lot of people who do networks often will map them to this sort of space. We did some conformal mappings with Drosophila embryos, or Drosophila imaginal discs when we were analyzing those data. So that's a conformal map is a type of hyperbolic space. And yeah, so this is nice. Be able to do this. If you can get that working, it'd be pretty good.
Yeah, sure. I'm like working towards it. I'm first figuring out the math of it because this is something new to me. So yeah, I'm trying out the math. I'll try to get it working.
Yeah. Okay.
Yeah.
Well, thank you.
Okay. Any questions, or I'll share the screen?
I don't know. Any questions?
Okay. Hello, am I audible?
Yeah.
Okay, so like in the first paper, you explained something about like interpretability. So do you plan to incorporate that in any of your like how would you like analyze that? I'm interested in this.
Okay, yeah, sure. So regarding what I said in the context of interpretability was that basically this paper which the idea which they're presenting in this paper is a simple geometric driven idea that I can see exactly if I have a certain structure, I can go back and see which neuron pulled this particular neuron so that it resulted in this structure. Since I have a fixed formula to calculate everything, so I can backtrack in my model and see what what attraction forces and all resulted in formation of say the middle circle I'm talking about. So what attraction forces resulted in forming this. So this model is completely based on it's not at all AI. It's not at all it's not at all machine learning or anything. So that's why it's interpretable. Is the point. The interpretability I didn't mean in a machine learning point of view. I meant
Okay, so it's not something about hallucinations. Like it's everything is calculated, so you just want to try
everything is calculated from basic formulas. So that's why it is interpretable. I can reason as to why this particular structure was made. That's what I mean from interpretability.
All right, got it. Thank you.
I did want to talk about Susan's, the thing Susan posted. So this is the paper that Susan posted in the chat. This is Control of Tissue Flows in Embryo Geometry and Avian Gastrulation. So this is Matthias Serra and some other people on this paper. So this is from Nature Communications this year or last year. So this is a fairly new paper. And so it talks about some of the types of physical phenomena happening during avian gastrulation. So gastrulation is this early stage in embryogenesis where you get things like folding and other types of cell movements. And so this is something that we want to understand. We especially want to understand cells and tissues that undergo coordinated flows so that the body plan can be established. And of course, this means that we're going from a very nondescript set of cells that are dividing to the different parts of the body that are going to emerge from this. So different organs, the limbs, the head, the tail, etc. And so this is what we're interested in, sort of the physics of setting this up. So here we elucidate how the interplay between embryonic and extra-embryonic tissues affect the chick embryo size and shape. These two distinct geometric changes are each associated with dynamic curves across which trajectories separate. These are what they call kinematic repellers. I think they're using the dynamical systems model here for this. Through physical modeling and experimental manipulations of both embryonic and extra-embryonic tissues, we selectively eliminate either or both repellers in Model 1 experiments, revealing their mechanistic origins. We find that embryo size is affected by the competition between extraembryonic epiboly and embryonic myosin-driven contraction, which persists when mesoderm induction is blocked. So we have the three germ layers that are emerging also during gastrulation, and we're inducing this into mesoderm, endoderm, and ectoderm, and they're going to go on to form different organs, different tissues. Instead, the characteristic shape change from circular to pear-shaped arises from myosin-driven cell intercalations, and this is just where the cells mix in different ways in the mesoendoderm, irrespective of epiboly. These findings elucidate modular mechanisms controlling avian gastrulation flows and provide a mechanistic basis for the independent control of embryo size and shape during development. So this kind of goes through and talks about sort of the relevance of what they're doing. So morphogenesis requires spatiotemporal coordination of hundreds of thousands of cells to sculpt a developing embryo. Elucidating the principles of morphogenesis, spanning mechanisms from molecular to organismal scales, is a grand challenge for developmental biology and the physics of living systems. So this is where they're trying to maybe answer some of these questions. So they're working from cell tracking data, of course, which we can gain from microscopy data that's recorded in time sequence. So you get an image of a bunch of cells, you segment those cells in terms of their cell body, their nucleus, and then you find the centroid of each cell, and then you figure out what position that is relative to other cells, and figure out the positional changes of those cells over time. So you can look at things like position, but you can also look at things like velocity and other types of things relevant to like cell migrations and accelerations and transformations. So that's what we want to be able to do. We want to get these spatiotemporal velocities describing the collective motions of cells. And so, sometimes we can do this. We have the data, but sometimes we have to recognize it's very noisy. And so sometimes we have to kind of figure out ways to filter out this noise. And so we need to be able to find sort of the signal through the noise. And this is where they talk about identifying kinematic structures. So this is where you're looking at different structures in, say, a flow field, and they're talking about the kinematic structures, or the structures that are relevant to movement. And so this is, they give the example of an ocean surface velocity field. One might be interested in understanding where floating objects accumulate or separate without knowing the precise forces generating the flows. So we're trying to track objects as a series of points, draw lines between those points, and infer things like, flow fields, flow boundaries, etc. Now, if you've been around our group for a while, you know that we have this interest in Lagrangian coherent structures. And this is exactly what they're trying to do here with the embryo. And there's been some really interesting work on Lagrangian coherent structures in the context of embryos. Matthias Serra is one of the people who is advancing this. There are a couple other people. And so Lagrangian coherent structures are this dynamical systems approach that uses a Lagrangian to look at, the dispersion of those points in space and in time, and then give like some sort of parameter that describes the structures that emerge from that dispersion. So if you have a bunch of cells that start at a central point and they disperse through a column of water, they're diffusing, they can diffuse at random, and you can look at that and where they diffuse in some way that's ordered, and you can look at that and use this measurement of it's a dynamical systems measurement where you're measuring sort of the distance from the center and the clustering of points. And a lot of times people will describe this as a set of ridges or a set of other types of structures. And so that's how we can understand this in embryos. And so in Lagrangian coherent structures, there's a lot of math. And, they have this approach called the dynamical morphoskeleton, which is, I think I talked about this paper last year, where they kind of map out how this can be applied to embryos. And so Lagrangian coherent structures allow us to reveal where cells maximally converge or separate over a developmental time interval of interest. So we end up with these dynamic attractors and repellers. That's what they were talking about, kinematic repellers. So you get attractors where things tend to aggregate. You get repellers from which places disperse, and you can identify those. And they want to make sure that you understand that this is a dynamical systems phenomena, not necessarily a way to describe things like chemoattractants, although there is a kind of an interaction there that's interesting. But the point is that you're looking at, say, like cell migration, you're looking at just the the way in which these cells are moving. You're not interested in anything else, and you want to make a description of the sort of the outcomes of those movements and find the structure. You're not describing like chemoattraction. You're not describing um, other aspects, mechanical aspects of tissue formation beyond the kinematics. And so they've used dynamical morphoskeletons in chick, fruit fly, and zebrafish embryos, and they're able to sort of get a handle on some of these early footprints of known morphogenetic features. And so they're basing this paper on some of that previous work, and they point out that repellers in particular were previously undocumented despite their potential relevance in dynamic tissue patterning. So they focus on avian gastrulation, and this is a process in early avian development during which a flat sheet of approximately 60,000 cells breaks symmetry, we were talking about earlier. So it instead of being this undifferentiated mass of cells, there's structure that emerges in this sheet. So when the cells break symmetry, they set up the vertebrate body axes and form the three germ layers: endoderm, mesoderm, and ectoderm. So instead of being an undifferentiated mass of cells, now they form these three layers, which will go on to form other tissues. So endoderm will form a number of tissues, mesoderm will form another set of tissues, and ectoderm will form another set of tissues. And it's sort of restricting the fates of those cells involved in these symmetry breaking events. And so that's kind of where we start to get structure in the embryo. At the onset of gastrulation, avian embryos consist of a circular monolayer of suspended epithelial cells, the embryo proper, surrounded by an annulus of extraembryonic tissue. That's what they mean by extraembryonic, is this ring or the circle around it. And so this is where we have this differentiation spatially of different cell types. Initially, mesoendoderm precursor cells are in a sickle-shaped region in the posterior of the embryo proper. Over the next 15 hours, these mesoendodermal progenitors converge and extend along the midline, which is the center line of this embryo, undergoing individual cell ingression, forming the primitive streak. The primitive streak, of course, being the first structure that you see, anatomical structure. This convergent extension is driven by active intercalation, apical constriction, and ingression. So this is all kind of where you're getting these processes that sort of shape these early tissues and early tissue formation. These processes generate complex embryo-scale coordinated cell movements and macroscopic vortical flows in the embryo proper. Similarly, the extraembryonic area expands outward. This is what they refer to as epiboly, powered by the active edge cell crawling on the vitellin membrane. The DM provides a concise summary of the complex cell movements during avian gastrulation. We have one line attractor and two repellers, and it's going to be in figure 1b. So if we go down to figure 1, we see in B, we see this an image here. We see in the image here, we have the extraembryonic area, the embryo proper, and we have the sickle-shaped area here. We have these two repeller regions and the attractor in the middle, which is the midline where we're going to get the primitive streak. And so this is where we get, so basically the mesoendoderm is going to shape into this, into the primitive streak. So that's what we're getting there. And then this is just showing intercalation and ingression, just showing the different movements of cells in this packing in the sheet.
I'd like to say something. It's interesting where the actin and myosin end up.
Yeah. In what way?
Well, they're in this repeller region, the blue.
Okay. Yeah, that's the domain of attraction. Yeah.
There's actin and myosin in that area that collects. There's another diagram of it. So.
Okay. Yeah.
Yeah, it's contractions.
Yeah.
Like contraction per se.
Yeah.
Yeah.
Yeah. And so, I mean, I guess I'll finish up by just saying a couple words about the way they're measuring this. So, yeah, and it says cyan in insets marked elevated myosin activity. So you get these, these different things happening along the edges of the cell here, or here, active constriction. So we need to constrict the cells as they move as well. So we need to account for all that. Now, this dynamic morphoskeleton, of course, is using this Lagrangian, so it's using a dynamical systems approach. It's describing all these movements with dynamical systems. You're tracking it. You can use, a discrete method of this. You can use a continuous method. So you can have you have to use a lot of calculus to kind of figure out the trajectory or you can use a discrete approximation of this as well, just tracking out these positions and then figuring out what the sort of the skeleton should look like, where the sort of the ridges are and things like that, which is useful. But it's kind of a interesting way of kind of approaching this problem. And so, the one thing we're doing here is we're describing kinematics, but we're not necessarily describing kinetics. So that's a different ball of wax. And, that's probably also important well, it's definitely important for cell movements and cellular physics. But right now we're just interested in the kinematics. Yeah, and then this just shows like the wild type gastrulation, some of the things going on. You go from this initial active stresses to this isotropic active stress, anisotropic active stress, and then they're looking at the flow velocities and divergence within these different states where you're getting these changes from this nondescript layering to where you start getting the primitive streak and you start getting this sort of organization of the different components of like the notochord and the nervous system and that. Yeah, so I think that's it. Thank you, Susan, for that paper. It was a good paper. Definitely interesting, and I'm not wanting to get any more into it. I think it's great. Do you have anything else to say, Susan? Or
Yeah, it's just that Dick was always looking for differentiation waves in other organisms other than the salamander, and this has some of the aspects of that because it's got actomyosin contractions and a concentration of actomyosin.
Yeah.
In certain areas. So if you go further down the paper, you'll see an image of that. But
okay.
yeah, so it was just something that I guess I could look at with regards to the actomyosin and certainly the epithelial cells involved. Anyway, it was just interesting.
Well, yeah, it was definitely interesting. Okay.
Somebody gave a slam talk on it.
Okay. Yeah. Yeah, it's good.
Yeah. Okay.
Right. All right. I think that's it for today. Thank you to our presenters, Shreyas and Hussain, and thank you, Susan, for bringing that paper up. And talk to you next week.
Okay.
Thank you.
Thank you. Bye.
Thank you. Bye.
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 Bye.